Over 1.5 million species of animals and over 500,000 species of plants are known. To deal with such a large collection of dissimilar forms, certainly we need some system by which species can be classified in a reasonable way. Many types of classifications are possible. We could, for example, classify flowering plants according to their color, height, or any other character. This type of classification is not meaningful, since it does not provide any information about the basic differences and similarities among different individuals. All organisms are related to one another at some point in their evolutionary histories. However, some organisms are more closely related to each other. Sparrows are more closely related to pigeons than either do the insects. Classification is based on relationship amongst individuals, that is, similarity in the form of structure. Biologists have classified all living things into groups showing similarities based on homologies, comparative biochemistry, cytology, and genetics. Large groups are divided into smaller groups up to species level. A species is a group of natural population which can interbreed freely among themselves and produce fertile offsprings. But are reproductively isolated from all other such groups in the nature. However, interbreeding cannot be used as a criteria for species recognition among predominantly asexually reproducing organisms. Each species possesses its own distinct structural, ecological, behavioral characteristics. Hence, species are independent evolutionary units. Different species do not exchange genes between them. Since long, the living things are divided into two kingdoms, plants and animals. Next, each kingdom is divided into smaller groups called phyla, also divisions for plants, algae, and fungi. A phylum, in turn, is divided into classes, classes into orders, and orders into families. A family contains related genera and genus, and a genus is composed of one or more species. Species is the basic unit of classification. Conversely speaking, the organisms are grouped into larger, more inclusive categories, or taxa. Each category is more general than the one below it, and hence emergent properties. The taxonomic categories from species to kingdom form a hierarchy. As described in the classifications of corn. Biological classification of corn, Zea mays. Kingdom Plantae, division or phylum Anthophyta or Trichiophyta. Class Angiospermae, order Poales, family Poaceae. Genus Zea, species Mays. Members of a lower category resemble one another more than do the members of higher taxon. From the earliest times, plants and animals have been given common names by the people. Since no system was used in choosing common names, in many cases, various regions had their own names for same plant or animal. Take onion for example. Its common Urdu name is piyaz, but in different regions of Pakistan, it's also known as ganda or wasal or wasal. In different countries, it would have another set of names. Similarly, amaltas, argwed, gurmala, golden shower, Pergincasia are common names for the same plant. Thus, the same plant may have different names. In some cases, a single name refers to several different plants or animals. What is bluebell? Dozens of plants with the bell-shaped flowers are called bluebells. Similarly, the word blackbird would means a crow as well as a raven. Common names have no scientific basis. To a biologist, a fish is a vertebrate animal with a backbone, fins, and gills. But silverfish is an insect. And a crayfish, jellyfish, and starfish do not fit the biologist's definition of a fish. Common names had long caused confusion. During the 18th century, Carolus Linnaeus, a Swedish botanist, devised a system for naming and classifying all the organisms known to him. His system is used today internationally. He discarded the common names of plants and gave each one a scientific name. He took the scientific name from Latin word. Linnaeus published the list of the names of the plants in 1753. The scientific names of each plant had two parts. Usually the name referred to some characteristics of the individual or a person who collected it. His system spread rapidly and became so popular that he used it later in naming animals and published his list in 1758. Many of his names are in use today. Linnaeus' system of giving each species a scientific name comprising two words is known as binomial nomenclature. The first name refers to the genus and is called generic name and always begins with the capital letter. The specific name follows the generic name and begins with the small letter. Scientific name for onion is Allium cepa. For amalthus, Casia fistula, and for man, Homo sapiens. Botanical name for potato is Solanum tuberosum, and for brinjal, Solanum melangina. The same generic name for the potato and brinjal reflects close relationship between these two species. Every species has its own specific name and world over. Initially, the classification was based on the appearance or morphology of plants and animals, but with advancement in the knowledge of cytology, physiology, genetics, molecular biology, the classification of organisms has been modified. For centuries, the living organisms have been classified into two kingdoms, plants and animals. Plants can prepare their own food from simple inorganic material and store energy. So are autotrophs. While animals cannot synthesize their own food from simple inorganic material and depend on their food either on autotrophs or on decaying organic matter, so are called heterotrophs. Bacteria were included in the plants. Many biologists found this system satisfactory, while others found it unworkable for many unicellular organisms. Like euglena, that have both plant-like presence of chlorophyll and animal-like lack of cell wall characters. And also because it ignores the differences between prokaryotic and eukaryotic cells. In 1866, Ernest Haeckel proposed a third kingdom Protista to accommodate euglena-like organisms and bacteria. In 1937, E. Chatton suggested differentiating terms prokaryotic, pro meaning before and karyon meaning nucleus, used to describe bacteria and blue-green algae. And the term eukaryotic, from Greek eu mean true, to describe animal and plant cells. Some biologists also disagree between the classification of fungi, such as bread mold, yeast, and mushrooms, which resemble plants in many ways but are not autotrophs. Fungi are a special form of heterotrophs that obtain energy and structural material by breaking down or decomposing and absorbing food substances from the surroundings, and possesses chitin, a major structural component in their cell walls. A relatively recent system of classification, the Five Kingdom system, was proposed by Robert Whittaker in 1969. This system of classification is based on three different levels of cellular organizations associated with three principal modes of nutrition: photosynthesis, absorption, and ingestion. The Five Kingdom proposed: number 1, the prokaryotic unicellular organisms, such as bacteria. Number 2. The eukaryotic predominantly unicellular organisms such as euglena and amoeba. Number 3. The eukaryotic multicellular autotrophs, plantae. Number 4. The eukaryotic multicellular reducers, for example, mushrooms. Number 5. The eukaryotic multicellular consumers. Plants are autotrophic in nutritional mode, making their own food by photosynthesis, such as mosses, ferns, flowering plants. Fungi are heterotrophic organisms that are absorptive in their nutritional mode. Most fungi are decomposers that live on organic material, secrete digestive enzymes, and absorb small organic molecules, which are produced by digestion. Animals live mostly by ingesting food and digesting it within specialized cavities. They lack cellulose and show movements, for example, birds and reptiles. In 5-kingdom classification, all eukaryotes that did not fit in the definition of the plants, fungi, or animalia were included in Protista. But this kingdom also includes relatively simple multicellular organisms that are believed to be direct descendants of unicellular protists. Lynn Margulis and Carlin Schwartz in 1988 modified 5-kingdom classification of Whitaker by considering cellular organization, mode of nutrition, cytology, genetics, and organelles of symbiotic origin, mitochondria and chloroplast. These 5 kingdoms are Prokaryote or Monera, Protista, Plantae, Animalia, and Fungi. About a century ago, at the time of Louis Pasteur and Robert Koch, the word virus was generally referred to as a poison. The present notion of virus is entirely different. Now, viruses are recognized as particles of nucleic acid, often with a protein code. They replicate in living cells and cause many diseases, such as influenza, hepatitis, smallpox, and AIDS. The branch which deals with the study of viruses is known as virology. The word virus is derived from Latin word venom, meaning poisonous fluid. It can be defined as non-cellular infectious entities, which contain either RNA or DNA, normally encased in proteinaceous coat, and reproduce only in living cells. Viruses utilize the biosynthetic machinery of the host for its own synthesis and then transfer themselves efficiently to other cells. Some viral diseases have been known for centuries. In fact, the first infectious disease against which effective method of prevention was developed was a viral disease. In 1796, Edward Jenner first vaccinated an 8-year-old boy with a material removed from cowpox lesion on the hand of a milkmaid. After 6 weeks, the boy was inoculated with the pus from a smallpox victim, but he did not develop the disease. Jenner used material for vaccination from cowpox lesions and successfully vaccinated 23 persons. As the material he used was obtained from cow, Latin vacca. Later, the term vaccination was used by Louis Pasteur for inoculation against disease. In 1884, one of the Pasteur's associates, Charles Chamberlain, found that bacteria cannot pass through porcelain filters, while agents responsible for rabies, a disease which is transferred to humans by bites of rabid dogs, foxes, cats, bats, and other animals, can pass through these filters. As in those days, the word virus was loosely used to describe any toxic substance that caused disease. Those unseen, filterable agents of the disease were described as filterable viruses. In 1892, Ivanovsky discovered that the agent which caused tobacco mosaic disease was filterable. He obtained bacteria-free filtrate from ground-up affected plants and placed it on healthy leaves of tobacco. He observed that filtrate produced the disease in healthy plants. Presence of similar filter-passing ultramicroscopic agents was seen in the victims of many diseases, including foot and mouth disease in 1898. The filterable agents were first purified in 1935 when Stanley was successful in crystallizing the tobacco mosaic virus. Chemical analysis of these particles showed that they contained only nucleic acid and proteins. This suggested that unlike other forms, viruses are of simple chemical composition. Characteristics: viruses are extremely small infectious agents which can only be seen under an electron microscope. They range in size from 250 nanometer of pox viruses to 20 nanometer of parvoviruses. They are 10 to 1000 times smaller than most bacteria. so they can pass through the pores of the filter from which the bacteria cannot pass. Viruses cannot be grown on artificial media. They can reproduce only in the animals and plant cells or in the microorganisms, where they reproduce by replication, a process by which many copies or replicas of viruses are formed. Thus, the viruses are obligate intracellular parasites. Viruses lack metabolic machinery for the synthesis of their own nucleic acid and protein. They depend on host cells to carry out these vital functions. During reproduction in the host cells, viruses may cause disease. All viruses are generally resistant to broad range of available antibiotics, such as penicillin, streptomycin, and others. The complete, mature, and infectious particle is known as virion. The virions are composed of a central core of nucleic acid, either DNA or RNA, which is also known as genome, and is surrounded by protein coat, the capsid. Capsid gives definite shape to virion. It is made up of protein subunits known as capsomeres. The number of capsomeres is characteristics of a particular virus. For example, 162 capsomeres are present in a capsid of herpes virus and 252 in capsid of adenovirus, which cause some common colds. In some animal viruses, the nucleocapsid, that is, nucleic acid and capsid, is covered by another membrane derived from the host, the envelope. Non-enveloped viruses are known as naked virions. Animal and plant viruses may be polyhedron, having many sides, helical or spiral. Edges occur in two structural forms, having cubical or helical symmetry. In general appearance, cubical phages are regular solid or icosahedral, having 20 faces, and helical phages are rod-shaped. Many phages consist of head and tail. In those cases, heads are polyhedral, but the tails are rod-shaped. Classification of viruses. Virus morphology and nucleic acid properties are most important for classifying plant, animal, and bacterial viruses. The genetic material may be DNA or RNA naked, enveloped, or complex. On the basis of morphology, viruses are classified into rod-shaped, TMV, spherical, polioviruses, and tadpole-like bacteriophages. Bacteriophages, viruses that infect bacteria, were discovered independently by Twort in 1915 and D. Herle in 1917. Twort observed that bacterial colonies sometimes undergo lysis, dissolved and disappeared, and that this lysis can be transferred from one colony to other. Even highly diluted material from the lysed colony can transfer the lytic effect. However, heating the filtrate destroyed its lytic properties. From these observations, he concluded that lytic agent might be a virus. D. Harley rediscovered this phenomena in 1917 and used word bacteriophages, meaning bacteria eater. Life cycle of bacteriophages. Earlier, researches on bacteriophages were mainly on limited number of phages. That infect E. coli. Of these, the best known phages are T phages. Among T phages, the T2 and T4 phages are mainly used in phage studies. The overall structure of T4 studied with the electron microscope resembles that of tadpole, consisting of head and tail. The head is an elongated pyramidal, having two triangular structures with a common base. Within the head, double-stranded DNA molecule is present. The structure of phage tail is more complex than the head. A layer of distinct proteins form the inner tube or core, which is enclosed in the sheath, made of another type of protein. On one side of sheath is collar, and on the other side is end plate. To end plate, six-tailed fibers are attached, which are structures for attachment. The volume of the phage is about 1 to 1000 of the host. The bacteriophage replicates only inside the bacterial cell. The first step in replication of bacteriophage is its attachment or adsorption to host cell at receptor site on the cell wall of bacterium. During attachment, weak chemical union between virion and receptor site takes place. In the next step, penetration, the tail releases the enzyme lysozyme to dissolve a portion of the bacterial cell wall. The tail sheath contracts and tail core is forced into the cell through cell wall. The virus injects its DNA into the cell just as the syringe. The protein coat, which forms the phage head and tail, structure of the virus remained outside the cell. Many animal viruses, however, enter the host cell as a whole. Immediately after entering the cell, the viral nucleic acid takes the control of biosynthetic machinery and induces the host cell to synthesize necessary viral components, DNA and proteins, and starts multiplying. About 25 minutes after initial infection, approximately 200 new bacteriophages are formed. Bacterial cell bursts. It undergoes lysis. Newly formed phages are released to infect the bacteria, and another cycle, the lytic cycle, begins. The phage which causes lysis of the host cell is known as lytic or virulent phage. All infections of bacterial cells by phages do not result in lysis. In some cases, viral DNA, instead of taking over the control of host machinery, becomes incorporated into the bacterial chromosome. Phage in this state is called prophage, and the process is known as lysogeny. In this condition, the bacterium continues to live and reproduce normally. Viral DNA, being the part of bacterial chromosome, passes to each daughter cell in the successive generations. Sometimes, however, the viral DNA gets detached from the host chromosome and lytic cycle starts. This process is called induction. Lysogenic bacteria are resistant to infections by the same or related phages. The phage which causes lysogeny is called temperate or lysogenic phage. The most recently discovered and the least understood microorganisms are prions, which may be infectious proteins. Their nature is very controversial. They are composed of protein only that contains the information that codes for its own replication. All other organisms contain their genetic information in nucleic acids, DNA or RNA. Prions are responsible for mad cow infection and mysterious brain infection in men. Smallpox. Smallpox, which is caused by pox viruses, the DNA-enveloped virus, is an ancient disease that is known to have occurred as an epidemic in China as early as 12th century BC. Until the early 20th century, smallpox was a common disease throughout the world. In smallpox, raised fluid-filled vesicles are formed, which become pustules later on and form pitted scars. By 1950s, immunizations and other control measures had largely decreased the danger, but it's still present in the third world countries, where many people are affected. In 1980, it was declared by World Health Organization that smallpox has been eradicated from the world. Herpes simplex. Herpes virus is a DNA virus. It's responsible for this disease. It's naturally occurring disease of mankind. In this, vascular lesions in the epithelial layers of ectodermal tissues are formed. Most commonly, this disease occurs in mouth, on lips, and at other skin sites. Influenza. Influenza viruses are enveloped RNA viruses. Influenza is widespread disease in men and occurs in epidemic form. Mumps and measles. Mumps and measles viruses belong to group paramyxoviruses. They are large enveloped RNA viruses. Mumps is highly contagious, widespread, but seldom fatal. About 60% of adults are immune to it. Measles is one of the commonest diseases of childhood, and adult human population is equally susceptible the world over. This disease develops immunity in its victim. Polio. Poliomyelitis caused by poliovirus is found all over the world. It occurs mostly in childhood. The age at which primary infection occurs varies with the social and economic factors. The polioviruses are the smallest known viruses and contain RNA in spherical capsid. retroviruses. RNA tumor viruses have been known for many years. These viruses are widely distributed in nature and are associated with tumor production in a number of animal species, such as fowls, rodents, and cats. Most familiar of viruses is human immunodeficiency virus, HIV, which causes acquired immune deficiency syndrome or AIDS. The single-stranded RNA tumor viruses, which also include retroviruses or oncoviruses, are spherical in form, about 100 nanometer in diameter, and enveloped by host plasma membrane. Although a few retroviruses are non-specific, that is, they can infect any cell, most of them can infect only host cells that possess required receptors. In the case of AIDS virus, host cell possesses a receptor that allows the viral adsorption and penetration in several types of leukocytes. In several types of leukocytes and tissue cells, the retroviruses have a specific enzymes called reverse transcriptase, which can convert a single-stranded RNA genome into double-stranded viral DNA. Not only this DNA can infect host cells, but it also can be incorporated into host genome as a provirus that can be passed on to progeny cells. In this way, some of the retroviruses can convert normal cells into cancer cells. Acquired immune deficiency syndrome or AIDS. The AIDS was reported by some physicians in early 1980s, having one or more of complex symptoms such as severe pneumonia, a rare vascular cancer, sudden weight loss, swollen lymph nodes, and a general loss of immune function. In 1984, the agent causing the disease was identified by research teams from Pasteur Institute in France and National Institute of Health in USA. In 1986, the virus was named as Human Immunodeficiency Virus (HIV). The major cell affected by HIV is helper T lymphocytes, which is a major component of immune system. As the HIV infection continues in the host, the decrease of the helper T lymphocytes results in the failure of immune system, and the infected person becomes susceptible to other diseases. Cells in central nervous system can also be infected by HIV. Recent studies on HIV reveal that the virus infects and multiplies in monkey, but does not cause disease in them, which means that HIV is host-specific. Avoiding the direct contact with HIV is an important measure for preventing the disease. Prevention of intravenous drugs with the common syringes and use of sterile needles or syringes and utensils is important. Now vaccine against HIV has been synthesized and its experimental administration in humans has been started in early 2001 in South Africa.